Good evening to Prof. Azhar and everyone. Today we'll be presenting on the CRISPR-based biosensor, which is a cutting-edge technology that has transitioned from the gene editing to revolutionizing molecular diagnostics. So our group consists of 3 members, which are SD, Helmi, and me. So I first start with the introduction. So CRISPR technology was first discovered as an defense mechanism in bacteria. So it gained a prominence in genome editing due to its ability to target the specific DNA sequences with high precision using the guide RNA. But its potential doesn't stop there. It's now being utilized in biosensors for detecting diseases. So in 2012, the CRISPR-Cas9 system was introduced for gene editing, and by 2016 the researchers adapted this system for the nucleic acid detection, opening doors to various applications like infectious disease diagnostics, the cancer biomarker identification, and even the environmental monitoring. So there are 3 main enzymes are used in the CRISPR-based biosensors. The first is the Cas9, which targets the DNA and introduces a precise double-strand breaks, and Cas12 also targets DNA, but it has an additional trans-cleavage activity that amplifies the detection signals, and Cas13 focuses on the RNA, making it ideally for detecting the RNA viruses such as the SARS-CoV-2. Now, looking at the general working principles, so CRISPR-based biosensors, it operates through 3 steps. Firstly is the target recognition by the guide RNA directs the Cas enzyme into a specific nucleic acid sequence, and second is the signal generation occurs when the enzyme cleaves the target sequence, producing a detectable signal such as the fluorescence. Thirdly is to enhance the sensitivity. Amplifications techniques like the PCR, they are often integrated into the process. So from the figure here, in the CRISPR-based biosensor, the CRISPR RNA in the CRISPR-Cas system, such as the Cas9, Cas12a, Cas13a, they can specifically biologically recognize the target, while the Cas protein is used for the cleavage of the target and reported after the recognition. So the biological signals they generated by the cleavage could be converted into other measurable signals and then read out by the fluorescence method, electrochemical method, the SIRS method, and the colorimetric method. Now I'll pass the floor to SD to continue with the recent advances in CRISPR biosensing technologies. Thank you.
Okay, I will be discussing about recent advance in CRISPR biosensing technologies. Let's start with electrochemical CRISPR biosensors. These biosensors are highly sensitive due to their ability to integrate CRISPR-Cas proteins like Cas12. When these proteins target and cleave nucleic acids, they amplify the signal, allowing us to detect even low concentrations targets. This process eliminates the need for PCR amplifications, making it more efficient. As you can see in the figure, the principle behind the electrochemical biosensors is based on detecting electrical signals produced when the CRISPR system cleaves specific nucleic acid. This is why electrochemical sensors are gaining traction. They offer high specificity and sensitivity. For example, the eCRISPR system, which uses Cas12a, which can detect specific DNA sequences using electrochemical signals. The use of square wave voltammetry (SWV) and methylene blue as a signaling agent allows for the detection of viruses like HPV-16. Next, let's explore next generation's point of care CRISPR biosensors. So these systems are revolutionizing diagnostics by being portable, rapid, and highly sensitive. They help overcome the limitations of traditional lab-based diagnostics, especially in remote areas or during disease outbreaks. One of the big advantages is their integrations with other technologies such as microfluidics and lateral flow assays. In the figure, You can see how different signal detection methods like fluorescence, polarimetric, and electrochemical sensing work. These methods are integrated into point-of-care CRISPR biosensors to allow for quick and accurate diagnostics in a compact format, which is crucial in urgent healthcare settings. Moreover, the integration with smartphones is being developed, enabling real-time data analysis and increase the accessibility, which you can also observe in the figure. Finally, CRISPR-based nucleic acid biosensors have become essentials in detecting DNA and RNA, which is crucial for diagnosing infectious diseases and genetic disorders. Biosensors are particularly important because they offer high sensitivity and specificity in detecting pathogens and mutations. As shown in the figure, the CRISPR-Cas system is effectively combined with nucleic acid amplification methods and various signal output strategies to improve diagnostic efficiency. This figure provides an overview of how these technologies work together in food safety and other applications, emphasizing their broad utility. So the integrations of CRISPR with isothermal amplification technologies like LAMP and PCR has significantly improved detection sensitivity and reduced time to results. As you can see, the potential for these biosensors to be adapted to various diagnostic needs is immense.
Moving on to the applications of CRISPR in medical diagnostics. So the CRISPR-based biosensors are particularly effective for diagnosing infections and the cancer due to their precision and speed. So I have reviewed two papers demonstrating the CRISPR technology application in viral specifically for COVID-19 and HIV. So both papers, they demonstrated innovative CRISPR-based detection platforms. The COVID-19 study presents an electrochemical APTA sensor integrating the CRISPR Cas12a technology, while the HIV study introduced a unique CRISPR gel platform combining the real-time reverse transcription recombinase polymerase amplification, the RT-RPA, with the CRISPR-Cas12a gene in an agarose gel matrix. So the technical performance of both systems, they are remarkable. And the COVID-19 APTA sensor achieves detection limits in picograms per milliliter, while the HIV platform, they can detect as few as 30 RNA copies. And both systems, they provide results within 30 minutes, significantly faster than the traditional methods. Also, clinical validation has demonstrated the excellent real-world applicability. So the both platforms, they have successfully detected their targets in clinical samples, with the HIV system showing a better performance compared to the standard RT-PCR methods. And these results highlight their potential for the point-of-care diagnostics, especially in the research-limited settings. So the technological advantages, they are significant. So both systems, they uses simple one-step processes requiring minimal equipment and expertise. So the COVID-19 platform uses innovative electrochemical detection, while the HIV systems using a gel-based approach provides the spatial separation of reactions. Next is the bacterial detection using the CRISPR technology. So the paper I reviewed focuses on the Staphylococcus aureus, which is a significant foodborne pathogen that demands the rapid and accurate detection methods. And this approach combines CRISPR Cas12a technology as well, but with the quantum dots in a lateral flow biosensor format, and the system works by using the Cas12a's transcleavage activity and triggered by a specific target DNA sequence from the Staphylococcus aureus. So when combined with the recombinase assisted amplification, this method achieves a remarkable sensitivity by detecting as little as 75 attomolar, which is an extremely low concentration of genomic DNA, or 540 bacterial cells per milliliter. So what are the key advantages? So it includes a rapid 70-minute detection time, a visual readout capability, and a high specificity with no cross-reactivity. This method has been successfully validated using the real food samples, demonstrating its practical applicability in food safety testing and the clinical diagnostics. So this platform represents a significant advancement in the point of care bacteria detection by offering, similarly to I mentioned just now, a cost-effective, user-friendly solution that could be adapted for detecting various pathogens. So its combination of the sensitivity, speed, and the simplicity makes it particularly valuable for the few applications and the resource limited settings. Furthermore, CRISPR-based diagnostic platforms can also be used for the lung cancer detection. So this system combines from the paper I reviewed, this system combines the CRISPR-Cas12a technology as well with DNA dumbbell probes and rolling cycle transcription to simultaneously detect two critical lung cancer biomarkers. First is the human 8-oxoguanine DNA and glycosylase 1, which is known as HOGG1, and the flap structure specific endonuclease 1, which is we can call it as FEN1. So the platform operates through a mechanism where the target enzymes it trigger a cascade of the molecular events leading to detectable fluorescence signals. And what sets this system apart is its self-recruiting CRISPR RNA design, which eliminates the need for the pre-assembled complexes and significantly reduces the background noise. Not only that, in clinical testing, the platform has shown a remarkable accuracy with recovery rates above 90% in both human serum and the lung cancer cell samples. So this high performance combined with its ability to simultaneously detect multiple biomarkers makes it a powerful tool for cancer diagnostics. So the system's versatility and the sensitivity represents a significant advance in cancer diagnostics by offering a potential applications in early detection, treatment monitoring, and also the drug development. In addition, now we look at the comparison between the CRISPR-based biosensors with the traditional diagnostic methods. So as you know, the traditional diagnostic plays a crucial role, but the CRISPR technology uh, advantages. So first is the specificity. So the CRISPR biosensors, they are highly specific due to their programmable guide RNAs, which can be designed to target a unique sequence and avoid the background noise from related molecules. And also the sensitivity is high, which is crucial for early detection when the target molecules, they are still small in amounts. Not only that, the CRISPR approach enables the multiplexing capability where the multiple biomarkers they can be detected in just only one assay, and it will potentially translate into a lower cost due to the simple designs and ease of use. So in short, the CRISPR biosensors, it contain the immense potential for diagnostics, particularly in situations demanding rapid, sensitive, and specific results. And also it provides a faster detection, and most importantly, it improves the patient outcomes. So for my small reflections on the papers that I have reviewed, I can say that recent advances in the CRISPR-based diagnostic platforms, they have revolutionized molecular detection across multiple disease types. And by leveraging the precise targeting capabilities of the CRISPR systems, particularly Cas12a, so the researchers, they actually have developed highly sensitive and specific detection methods for viruses such as the HIV and COVID-19, the bacterial infections, and the cancer biomarkers. And what makes these developments particularly good is their versatility and the adaptability. So I can see the innovative approaches ranging from the electrochemical sensors to fluorescence-based detection and simplify one-pot system like the CRISPR gel. And these platforms, they are breaking a new ground in making the molecular diagnostics more accessible and user-friendly. Also, another standout features is the ability to perform the multiplex detections, which allows the simultaneous monitoring of multiple disease markers. And this is particularly valuable in complex conditions such as the cancer, where the multiple biomarkers, they can provide a more complete diagnostic pictures. However, there are still some challenges remain in the standardization and also the clinical integration. So the field is actually rapidly moving toward the point of care applications that could dramatically improve the disease detection and also the monitoring. Therefore, the combination of the high sensitivity, rapid results, and the simplified workflows, it places the CRISPR-based diagnostics as a transformative technology in the modern medicine. So I can see that these developments, they suggest a future where the rapid, accurate molecular diagnostic become readily available in the various healthcare settings, potentially revolutionizing how we approach the disease detection and monitoring. So the continued evolution of these platforms, they can make promises to make the molecular diagnostics more accessible and practical for routine clinical use. So that's all from me. Thank you. Now I'll pass the floor to Helmi to continue with the presentation.
Next, I will talk about the challenges of developing a CRISPR biosensor. Different CRISPR systems have different Cas proteins, which can flexibly selected for different target characteristics and have the advantages of high detection sensitivity and specificity, which is an important development direction in the field of rapid detection at the current situation. The use of the CRISPR-Cas system in the construction of nucleic acid biosensors enables a perfect combination of the sensitivity, specificity, and flexibility of CRISPR-Cas with the simplicity, timeliness, and the low cost of nucleic acid analysis. Through the integration of these new technologies, it is possible to quickly and accurately assess various risk factors in food market supervision and effectively stop the problematic food from entering the market. In this figure, it is an example of the CRISPR-Cas system biosensor in the food analysis. The blue part indicates nucleic acid amplification methods. The orange part indicates signal output analytical system, and the green part indicates the food analysis target. CRISPR-based biosensors have revolutionized molecular diagnostic by providing high specificity, sensitivity, and rapid detection of nucleic acids. These biosensors leverage CRISPR-associated proteins such as Cas9, Cas12, and Cas13 to detect DNA and RNA sequences in diverse applications, including disease diagnostic, environmental monitoring, and food safety. The incorporation of aptamer expands the detection of targets of CRISPR-based biosensor to protein, small molecules, exosomes, and bacteria. Furthermore, this CRISPR-based biosensor can meet various detection requirements by employing different signal readout methods such as the fluorescence method, electrochemical methods, and colorimetric methods. However, there are still many challenges that remain for the development of CRISPR-based biosensor. Addressing this limitation and exploring future development will be crucial for the full-scale deployment of the CRISPR-based biosensor. First challenge is sensitivity and specificity. While CRISPR-based biosensors exhibit high specificity, their sensitivity can be affected by factors such as sample complexity, target concentration, and interference from non-specific sequences. Enhancing signal amplification strategies is necessary to improve the detection limits. The sensitivities of the CRISPR-based biosensors need to be improved by integrating more signal amplification method into the CRISPR-based biosensors that can improve the detection sensitivity, such as the isothermal amplification, biotechnologies, and signal amplification nanomaterials, such as gold nanoparticles, And reducing the background can also improve the sensing performance, which is generated by the long reaction time. Second is the delivery and sample preparation. Effective delivery of CRISPR reagents into target samples remains a challenge, especially for complex biological matrices such as blood or environmental samples. Efficient sample preparation techniques are needed to minimize the background noise and maximize the target availability. The third one is the cost and accessibility. Despite the potential of CRISPR-based biosensors require expensive reagents and sophisticated equipment, limiting their accessibility in resource-limited settings, developing a cost-effective alternative and portable diagnostic platform is very essential. Next is the discovery of Cas proteins. The discovery of the Cas proteins family is still incomplete, with many new Cas proteins yet to be identified. A researcher named Harrington discovered a Cas14 which can be used in bind and cleave the target of ssDNA. The Cas14 is more specific than Cas12a in recognizing ssDNA, showing the great potential in biosensing technology. The fifth challenge is detecting the non-nucleic acid target. The application of CRISPR-based biosensors for detecting non-nucleic acid targets still very limited due to the lack of a universal method. The aptamer is used to bind the non-nucleic acid target to signal conversion, but this approach is constrained by a narrow range of target. Last challenge is the storage of CRISPR-based biosensor. The storage of CRISPR-based biosensor is challenging because the protein used in the detection system cannot be stored at the room temperature. The use of a freeze-drying strategy can solve this problem, but the activity of thermal enzyme used for application might be very limited. Moving on the next part is the future perspective of CRISPR biosensor. This figure is a summarize of the future perspective in CRISPR-based biosensor that I will explain in the next slides. First future perspective is exploring potential properties of CRISPR case nucleases. The target recognition mechanism of the CRISPR-based detection mainly depends on the properties of the Cas nucleus. The emergence of artificial intelligence, particularly tools like AlphaFold, has facilitated the prediction and analysis of protein structures. By integrating this technology with protein engineering techniques, it becomes possible to modify Cas nucleases and explore their potential properties. These modifications can improve the properties of Cas nucleus, such as high temperature resistance, PAM site versatility, and high recognition accuracy. Therefore, the combination of artificial intelligence tools and protein engineering may open a new avenues for improving the customizing the functionalities of Cas nucleases, leading to sensitive, specific, and multiplex detection of infectious disease. Second future perspective of CRISPR-based biosensor is establishing a standardized sample pretreatment process. While several CRISPR-based biosensors have already been developed for the detection of infectious diseases, a major challenge is that there is no standardized procedure for sample pretreatment. Different pretreatment methods are required for infectious disease detection from environmental, food, and clinical samples. The lack of standardization makes it difficult to objectively compare the performance and reliability of different detection methods. Therefore, establishing a standardized sample pretreatment procedure is essential for accurate and consistent comparisons between the different CRISPR-based biosensors and to facilitate the development of rapid detection. The third future perspective of CRISPR-based biosensor is detecting non-nucleic acid targets. Currently, most CRISPR-based biosensor studies are centered around detecting nucleic acid targets. However, to expand the versatility of these biosensors, some researchers have focused on detecting non-nucleic acid targets, such as organic small molecules and proteins. A noteworthy challenge in developing activable CRISPR-based biosensors for non-nucleic acid targets is devising effective methods to translate the target recognition process into the collateral cleavage activity of Cas enzymes. In this context, aptamers and bacterial allosteric transcription factors ATFs emerge as potential solutions for enabling the detection of non-nucleic acid targets using the CRISPR system. By leveraging the unique properties of aptamers and ATFs, researchers can design innovative approaches to facilitate the non-nucleic acid target detection and expand the application of CRISPR-based biosensor. The fourth future perspective of CRISPR-based biosensor is developing amplification-free strategies. The incorporation of nucleic acid amplification methods can significantly improve the sensitivity of CRISPR-based biosensors, but they also pose the risk of non-specific amplification and potential cross-contamination. Although several CRISPR-based biosensors have been developed for the amplification-free detection of infectious diseases, it is important to note that many of these methods are still in the lab validation stage. This is mainly because the limited number of the targets in the real sample makes it difficult to achieve sensitive and reliable detection using the amplification-free CRISPR-based biosensors. Addressing this limitation is crucial for advancing the potential and practical of application of amplification-free CRISPR-based biosensor in the real-world scenario. The last future perspective of the CRISPR-based biosensor based on the review of the journals is the improving detection at a high throughput manner. With the ongoing mutation of the influenza viruses, the importance of high throughput detection is becoming increasingly prominent. However, the vast amount of data generated from high throughput assays presents a challenge in terms of data analysis. In this context, machine learning offers a promising solution for the effective analysis of such data. The use of machine learning algorithms can improve the accuracy of results, allowing discrimination between different mutant viruses. This application of machine learning in the analysis of high throughput assay data has the potential to improve our understanding of influenza virus mutation and contribute to more effective detection and surveillance strategies. Next future perspective of CRISPR-based biosensor is integrating CRISPR-based biosensors onto portable devices. To overcome the limitation of sophisticated instrumentation for detection, the CRISPR-based methods can be integrated into portable devices such as smartphones, portable electrochemical stations, and handheld Raman spectrometers. Most of these portable devices are equipped with their own batteries, which facilitate data collection and analysis directly in the field. Furthermore, the real-time uploading of data to the cloud through network connections enables infectious disease monitoring and facilitate the establishment of risk prediction models. Taking advantage of these portable devices, the accessibility and the applicability of the CRISPR-based biosensors can be expanded, allowing them to deployed in resource-limited settings and remote areas where sophisticated laboratory equipment is very limited. Last but not least, I will conclude this presentation for the CRISPR-based biosensor. In conclusion, CRISPR-based biosensors hold an immense potential for rapid, accurate, and portable diagnostic in medicine, food safety, and environmental monitoring. Overcoming current challenges through the technological advancement and strategic collaboration will pave the way for their widespread adoption. Future research should focus on improving insensitivity, affordability, and integration with digital platforms to harness the full potential of CRISPR-based biosensors in various sectors. Continued research in AI-driven protein engineering, high-throughput methods, and non-nucleic acid detection will drive the future innovations, making the CRISPR an indispensable tool in biotechnology and healthcare. That's all from us. Thank you. This is the last part of this presentation, which is the references. There are about 10 journals for our references for preparing these slides. That's all from us, Helmi, Fuher, and SD for this presentation of CRISPR-based biosensor. Thank you, Prof. Azhar.